Our next great discovery answered another question about the biology of life. In the late 19th century, biologists knew that during formation of the embryo, the cells of most organisms became differentiated. That is, they receive their assignments for specific jobs in the adult organism. Arms, legs, eyes. But when did this differentiation happen? German biologist Hans Driesch was determined to find the answer. In a lab experiment with sea urchins, he observed that in the early stages of an embryo's development, its cells are not yet differentiated. They can still develop into all cell types. Today, scientists call cells that can develop into all or many cell types stem cells.
Hi, Bill.
Hi.
To find out more, I paid a visit to Helen Blau, who is directing stem cell research at Stanford University's Genetic Pharmacology Lab.
Everybody's talking about stem cells. Cells start out, they're fertilized, then they start to divide, but they still haven't started to divide into what I would think is a liver cell, a fingernail cell, an eye cell, right?
That's right.
And what do you call that?
So at that point, they're totipotential. They have a lot of plasticity. They can give rise to all the cells of the organism. And then something changes. And we don't know what that something is, but those cells are now destined to make different kinds of tissues. And that's why we have organs such as the heart and the liver and the spleen in the adult. So then the question is, how irreversible are those decisions?
Okay.
So I wanted to challenge the idea that cells were irreversibly determined, that once a cell became liver, that was it. And so we showed that they were not irreversibly determined, and that by perturbing their environment inside the cell, you could activate genes that were usually never used. That demonstrated a certain kind of plasticity, and it's this kind of plasticity that people are trying to enlist in stem cells. So understanding the mechanisms underlying this may allow us to use the fundamental principles we've discovered in treating disease. What's recruiting these cells? What's getting them to fuse? What's getting them to reprogram so it's even more efficient? We might enlist that in what's called regenerative medicine. We're finding that cells have more potential than we thought before. So that a blood cell stem cell not only makes the blood, but those cells can travel elsewhere through the circulation to aid in the repair of damage in tissues such as muscle, tissues of the brain, also liver. And that's been shown in several labs now.
That's like Star Trek or something. You know what I mean? Dr. McCoy stabs the guy, and 15 seconds later, he's okay.
Well, it's not going to be quite like that.
Yeah,
we got a long way to go. But
To gain a better understanding of the new technologies that Helen Blau is using to uncover the potential of stem cells, she showed me an experiment being conducted with genetically altered mice.
Why not? Sure.
So that is one unusual mouse.
It certainly is. It has green fluorescent protein in all of the cells of its body. Because we've been able to introduce the jellyfish or GFP gene into these mice, now their tissues glow. And you can see that particularly in the ears, the eyes, the tail.
It's spooky.
But all the tissues are green. This has enabled us then to take cells that glow and put them into another mouse and see what tissues they participate in. So in a way you can follow what stem cells can do.
It's a marker.
It's a marker.
And it just, all you need is an ultraviolet light.
That's right. And it didn't exist before. And because of that, we now can follow the fates of these cells, where these cells go, what they become, in a way that wasn't previously possible.
It is fantastic.